welcome. Thank you all for coming. I'm going to talk about my international work today, and I'll start by saying that one of the advantages of traveling internationally is that I've been vaccinated for just about everything, so I can enjoy the cookies. If you have not had those vaccinations, then good luck to you. So, okay, my target audience today is primarily the students, particularly the American students, but I'm hoping that there'll be something in here for everybody. Here's my background. We've already gone over that. I'm a native of the Midwest, spent most of my life here. And my job is primarily extension. I provide pest management expertise to fruit and vegetable growers. It's a very rewarding job. I'll talk about that more in a few minutes. I also do applied research. I've done some teaching and do a fair amount of international work as well. So my international experience has been in 1997. I worked in a project in Ukraine on potatoes. For about 12 years, I worked in West Africa, worked on green beans, hibiscus, and tomatoes going to be the primary topic that I talk about today. Then I worked in Mexico on crucifers and tomatoes. Have done a lot of work in recent years in Afghanistan. You can see the list of things that we've been involved in there. And then for the last 3 or 4 years, been working in Honduras as well, primarily on zebra chip on potatoes. So some of you are old enough to remember the 1992 vice presidential debate when Ross Perot's running mate asked these two questions at the beginning of the debate because nobody knew who he was. Who am I and why am I here? And I think those are good questions that we all need to ask ourselves from time to time. And you students in particular, you've been blessed with incredible intellect. The fact that you're here tells us that you are intelligent and you have a great love of science. That's why you're doing the things that you do. And so why? Why have you been blessed in that way? What is your purpose? And so that's kind of what I want to get into today, not to get too philosophical or theological, but to talk about what becomes of your work, okay, and how can it touch people. And when I ask the question, why did I get involved in international work, and I will answer that throughout the talk today, I go back to the fact that the goals of extension are very similar to the goals of international work. The reason that we have the land grant system here in the United States is that the government provides funding for us so that we can help people, help our country, help the community at large in various ways. And one of the things that I enjoy doing in extension is helping people solve problems. That's one of my favorite things to do, and sometimes we do that with formal educational programs and those sort of things, but we also do that just with one-on-one conversations. And when a couple weeks ago I was at the Hort Congress down in Indianapolis, and we had about 900 growers there. I gave 5 presentations, and that all went well, but the best work I did was when I met people in the hallway and talked to them about their particular problems and how they could improve their operations through better management of their pest. And so the goals of extension very much line up with the goals of international work. The reason we do those things is try to help people primarily in developing countries to improve their livelihoods, improve their lives. From time to time, I've been asked to give presentations to various groups talking about how to develop an effective extension program. And I always begin those talks with a slide that says this: if you don't like people, find another job. Extension is a very people-oriented career. And if you don't like working with people, you don't like helping people, find something else to do, okay? And so the same would be true in international work. The reason we do it is to help people who need some of the knowledge that we have. Okay? If you think you might want to get involved in international work, there are some things to consider. A few years ago, I took one of my graduate students, Craig Robus, to Africa with me. And when we got back, I asked him, what was the main thing you learned there? And he said that when our major professors travel overseas, they're not going on vacation, okay? Because it's not like going on vacation. It's hard work. Adjusting to changes in time zones is hard. You may be put in situations that are uncomfortable. In our travels around West Africa, I ate things that I never dreamed that I would eat. I slept places that I never would have thought I would sleep. I took showers in places that I wouldn't normally walk into with boots on. So you get put in a lot of uncomfortable situations sometimes when you do international work. And then a real important consideration is you may be away from your family for a long time. I was fortunate. My wife was a stay-at-home mom, and so she could take care of the kids and keep the family going while I was gone. But the fact that I was gone did put strain on everybody. So that's a consideration when you start talking about doing international work. There are some technical considerations too. If you're used to American agriculture, you might find that the farming methods are very primitive. A lot of work is done by either animal traction or by hand. And so the farming methods aren't what we're used to. The farmers may have little or no education. A lot of our farmers now have a bachelor's degree at least. And so they're highly educated, they're very tech savvy and those sort of things. In a lot of the countries that we work in, that's not the case. It's likely that the country you're working in will have no functional extension system. They may have an extension system on paper, but it may not be functional. I was in Burkina Faso a few years ago, and they had a pretty good extension system set up, but the government cut the funding in half. And so I asked one of the agents, so does that mean you only have half as many extension agents as you used to have? And he said, no, we have all the same number of agents that we have, we just don't leave our office. Okay, well you can't do extension from your office, you have to go to the farms. So the goals of adoption of IPMs by farmers may be different than in the United States. In the United States, they're trying to increase their profit margins. Kind of maybe a basic thing would be stay in business. Increase profit margins, try to expand their operation, and those sort of things. In developing countries, their goals may be completely different. So if you're talking about subsistence agriculture, they just may be trying to grow enough food to survive. Okay? A very different mindset than what our farmers have. They use excess production to sell. And then they purchase things that we would consider necessity, things like education for their children, health care, and those sorts of things. They may want to expand or improve their operation. A lot of the farmers that I work with in Mali, and I'll talk about that later, they do all their irrigation by hand. And they have to irrigate all their plants twice a day. And so they may either draw that water from a well or dip it out of a river in a bucket. And then they take that and manually pour the water on every plant twice a day. And so some of the children, that is their entire job. That's what they do every day, all day, is they water all the plants in the morning, they go eat lunch, come back and they water all the plants again, and that's what they do. Well, we worked with one group in Kati, which is just outside of Bamako, and they use their profits from growing vegetables to buy a pump, okay, a simple gasoline pump. So they ran a hose to the river, and then they used the hoses to water the plants, and it reduced their labor tremendously. So now they can grow a much larger area of vegetables because they have water available. And then in a lot of cultures, if you make a lot of money, you get a second wife. Okay, this is not necessarily what we're hoping for, but that's the reality. And so the goals may be quite different depending on where you are. And then economics are different. They may have little or no access to capital. So if you say, well, you know, if you bought a tractor, you could really increase the area that you're farming. Well, if you make $600 a year, and you're not likely to be able to buy a tractor, okay? And and even ideas like buying hybrid seed instead of saving your own seeds may not be possible because there's no money available to do that. Labor is often very cheap. One of Mohammedi's colleagues in Herat told me the story when I was there that one of the ways they control grasshoppers and wheat is they give a bunch of guys a sweep net and they march across the field sweeping with the sweep net, and they collect the grasshoppers and feed it to the livestock, and so they're gathering animal feed and controlling their insects at the same time. So in the United States, obviously, we can't do that. You know, I talked to some of our fruit and vegetable growers. Their basic salary for migrant labor is about $15 an hour. And that's for migrant labor. And the reason they hire migrant laborers is Americans won't work for $15 an hour. So labor is not that cheap here, so it's very different. And then often their management skills are very lacking. They are doing things the way their ancestors have done them, and not much has changed, and they don't have a lot of management skills. Okay. The channels of trade may or may not exist to supply inputs. If you're close to a big city, you may be able to buy seeds or pesticides, but otherwise you may not have access to those things. Okay. But one thing I have noticed, no matter where I go in the world, they always find insecticides. Okay. Each one of these is a empty bottle of insecticide that they just piled next to their irrigation water. So somehow they always come up with insecticides. Now, there may not be any insecticide in the bottle because a lot of times there's no quality control on those sort of things, but they're buying it and they're spraying it. So they can always find insecticides, it appears. And the other thing about international work is you can't live there necessarily, so you have to rely on in-country collaborators, and some of those are very good and some of them are not so good, but you really don't have any choice about who you're going to work with. This is Madame Gambi. She was one of my collaborators in Mali, and this is Musa Nusaro. And I want to tell you a little bit of a story about Musa. Some of you probably remember he was here for a semester about 10 or so years ago. Musa, the first trip I made to Mali, I met him, and he was working on a flea beetle that was a pest of hibiscus, and he was studying a couple of predators that would eat the flea beetles, and he was trying to rear them together and get the flea beetles to, or the predators to feed on the flea beetles, and they wouldn't. And he was growing them inside a mud hut with one window and no electricity. And he couldn't figure out why they wouldn't feed. Okay, so I told him, okay, go outside, build a structure with just a roof over it to keep the rain off, and then rear them out in the sunlight. And when he did that, of course, they started feeding and everything was fine, and we got a lot of good data from his studies and so forth. But what I noticed about him was, even though he was working in very, very primitive conditions, no electricity, no running water, he had a keen understanding of biology. And so I kind of latched on to Musa. And later on, I became an adjunct professor at the University of Bamako, which is the capital city of Mali. And he got his master's degree there with me. And then later on, he got his PhD there. He was the first PhD graduate of the University of Bamako Department of Biology, and he graduated with highest honors. He went from raising or working in an environment where he did not even have electricity to coming to the United States, working at UC Davis doing molecular biology. So he really progressed in his career, and it was just a real fortunate coincidence that I ran into him on that first trip. And one of the things Musa always tells me every time I see him is that I changed his life. And the truth is I did not. He changed his life because he was such a hard-working person. And I'll talk about him more in a few minutes. What I'm going to do now is talk about a specific project that we did in West Africa where we looked at whitefly vector viruses on tomatoes. And if you don't know where Mali is, you probably haven't been watching the news lately. When I first started working there, I didn't know where it was. I had to look it up on a map. But here it is. Okay, it's a very large country. Our work there eventually took us to Burkina, to Niger, to Benin, Togo, and Ghana as well. So we worked in these 6 countries, but primarily we're working in Mali. Okay. Here's a closer map of Mali. The capital, Bamako, is down here in the south, and here's the Niger River. Okay, this is the primary source of irrigation water for the entire country. This is Timbuktu, and north of here is all the Sahara. There's just not much there except sand, and of course this is where the fighting is taking place now between the Islamists from Algeria and Libya and northern Mali and the government troops. Okay, and my student Musa, his family is from Gao, and that city was in the news recently because the Islamists took control of it, and now the French have expelled them, so that's where all the fighting is taking place. Mali is a very, very poor country. Only about 4% of the land is tillable. Okay, so a lot of it is not good. There's about 12 million or so people there. Okay, just a little history lesson. Mali was a French colony up until 1960 when it was granted its independence. It then became under the influence of the Soviet Union, and so they had a Marxist government. Many of the scientists were trained in the Soviet Union for many years. In the early 1990s, they had a coup, and they overthrew the government. They established a democratic form of government. They went through 2 election cycles while I was working there where they changed parties who ruled the country, and everything went smoothly until last year. And then things kind of went downhill. There was a military coup. It overthrew the government, and that allowed some of the tribes in the northern part of the country to take over that part of the country and kind of expel the government forces and so forth, and now it's just kind of a mess. Okay, so back to tomatoes. Tomatoes were an important crop in West Africa because they eat a lot of rice with some kind of a tomato sauce on it. That's a traditional meal. They produce those for fresh market sales in Mali and exported them to other countries. The excess were processed in a cannery, and they were a net exporter of tomatoes and tomato products. Okay. Many of the tomato growers are women. The men tend to grow the agronomic crops, and the women tend to grow the vegetable crops. Okay, so here we see a traditional farm. You can see the plots are very, very small. Okay, and so it would be very, very small pieces of land devoted to tomatoes. And then some of the other farms that we traveled to are pretty large acreages of tomatoes in one site. So there's a lot of diversity in how the farms operate. Almost all the work is done by hand, very little mechanization. Even preparing the soil, it's done with a hoe, not with either animal traction or with tractors. The distribution system is pretty primitive. This is how they take their tomatoes to market. And the market is primitive by our standards as well. And you can see the tomatoes here and some of the other vegetables and grains that they're selling at this market. When they have excess production, they dry their tomatoes. I'm not sure what our food safety people would say about drying it on the ground, but this is how they take care of their excess production, and then they save these for use later in the year. There was a cannery there. Here's the outside, and they have some equipment there. It's kind of old, but it functions fairly well, and would be profitable if it were still running. Now, here's the problem we ran into. About 2000, virus problems hit the tomato industry in West Africa and had great reductions in production. And the cannery closed because there was no consistent supply of tomatoes, and they had some bad management as well, but it was no longer in operation. And Mali then became a net importer of tomatoes. Okay. The virus symptoms that we saw appeared to be similar to tomato yellow leaf curl virus, which is vectored by sweet potato whiteflies. Okay. So what we did was we created a team to work on this problem. Bob Gilbertson was the virologist. He's from UC Davis. Musa was an entomologist there. Madam Gambi also there. The Sufu Kolo is a plant pathologist. He actually got his master's degree here at Purdue. He was a native of Niger, so he worked at AVRDC. And he worked with us. And then Jeff Gordon, before he became a stock car racer, was a plant breeder at Cornell University. So, and here's the team. Here's Bob Gilbertson, Madam Gambi, there's Musa in the back, there's Jeff Gordon there, and there's the Sufu. Okay, here are some of the symptoms that we saw. Okay, and you don't have to be a virologist or even a horticulturist to know that's not what tomatoes are supposed to look like. Okay, these are pretty ragged looking. Okay, more symptoms, and those are pretty ugly. And in this particular field here, it was a farm that was located right on the Niger River, and it was farmed by a woman. She had probably a half acre of tomatoes growing there, and every day she would walk down to the Niger River, get a bucket of water, and one by one water every one of those plants. When we were there, this is what they looked like. And with all that work she did and all that investment she had in that, she was going to get nothing. The symptoms were so bad that none of these tomatoes were going to ripen. She would never get any income from that at all. And it was heartbreaking to see this woman working so hard and knowing that she was going to get nothing from it. So beginning in 2003, we started taking samples of tomatoes and other crops that had virus-like symptoms. We brought them back to the United States, and we sequenced the DNA out at UC Davis and compared them with previously known viruses. And what we actually found was there was a group of 3 different viruses that were causing the problems. And one of them was indeed a yellow leaf curl virus, but it was unique enough that it was given the name tomato yellow leaf curl Mali virus because it was a new virus. Okay, and here's kind of the symptoms that we saw with that one. The most important one that we found, however, was tomato leaf curl Mali virus, which again was a unique virus, and those are the symptoms that we would see with that one. And then the final one we found was a leaf crumple virus, and there you see the symptoms on tomatoes and on peppers, and that was also a real important one as well. So we had this whole complex of viruses that were causing the problems. Now, one of the things we needed to find out was what host plants would harbor the virus. And so Bob did his DNA stuff that I don't fully understand, but he does. And we found that tomatoes were indeed the best host, followed by peppers. And one of the other big crops that they have is African eggplant, and it did not harbor the virus at all, which was really good. We were not able to identify any weeds that were hosts for the virus, although it's possible that they were carrying it, just not in sufficient levels to be able to detect. And all this is really important. We're going to talk about the host-free period in a few minutes, but and I'll get to that in a little bit. We also need to look at the vector, the sweet potato whitefly. And what are the hosts for that? All the solanaceous crops are hosts, cotton, which is an important crop, cucurbits, okra, lots of crops, lots of weeds. The bottom line was they fed on just about everything. Okay, so all during the year, it didn't matter when it was, there were sufficient hosts there for the whitefly to be present in pretty decent numbers. So our focus then had to be more on the virus than on the whitefly, because we couldn't get rid of the whitefly. So one of the things we wanted to do then was look at germplasm to see if there are sources of resistance available. And so we looked at a number of different varieties. We put out small plots in a number of different countries to see how they fared. And then after we identified the ones that had some resistance, then we wanted to distribute seeds to growers for large-scale testing. And just an example, one of the research plots that we had. And here are some of the results. This is just a sample. Roma was the standard variety. Okay, you can see that depending on the year, you could lose all the plants to virus. It was that susceptible. And so we looked at a number of different varieties. Gem Pride is one that looked really good in terms of virus resistance. The problem was these are what we would call canning type varieties, Roma types. Gem Pride is a fresh market variety, so the growers really didn't like that one because it didn't ship well and didn't have enough soluble solids and those sort of things. So they really didn't like that, although it was obviously good. So these Heinz varieties were really acceptable to the growers. They liked those varieties. They liked Mongol, and all of those had pretty good resistance to diseases, even though not as good as the Gem Pride. When we looked at yields, again, this Mongol was a really good yielder, and it had moderate levels of resistance, so that was one that had a lot of promise. These Heinz varieties also looked really good. The other thing we should point out here is that 2005, 2006, we had all this bacterial wilt problem. So you never operate in a vacuum where you just got one problem to deal with. There's always other stuff coming along that messes things up. But we did. We're able to figure out that these are some of the varieties that we need to look further at and test in large plots. Now, back to the insects. The whiteflies are present all year round. This is the sweet potato whitefly. This is a greenhouse whitefly. Their life cycle is about 30 days, and they can be very mobile. Okay, so they can travel from field to field, from plant to plant within a field, very, very readily. And when you're talking about virus transmission, that's really important because they got to pick up the virus somewhere, and then when they feed, they infect the plants that they feed on. You can see here, sometimes there are a lot of whiteflies there. Okay, now, in terms of transmission, as far as we know, only the sweet potato whitefly will transmit the viruses. And there's no transovarial transmission, so it doesn't go from one generation to the next. And so therefore, every new generation is clean. Okay, there's no virus there, so they have to pick that up by feeding on an infected plant. The acquisition period is very short, and once they acquire the virus, they're pretty much viruliferous for the rest of their life. And as few as 15 whiteflies that are carrying the virus per plant results in 100% infection. Okay, so it's not hard to get 15 whiteflies per plant. And then the diseases are expressed in about 15 days. The early infected plants don't produce hardly any yield at all. So timing is really critical. If we can keep the whiteflies away or keep the virus away until the plants get older, then we can get some yield out of there. And later infection may cause the flowers to drop off or fruit not to ripen properly. So, but the earlier the inoculation, the more severe the impact on yield. So, you know, as an entomologist, one of the things we look at is, okay, well, maybe we can control the vector with insecticides, and then we won't have a problem. We've got two approaches. We can use systemic insecticides on young plants, or we can use foliar insecticides later on. But the problem, of course, with insecticides is when you're dealing with a vector that can very rapidly transmit that virus to the plant, is even if the insect lands on there, starts feeding and dies, the plant's already infected, so it may not do any good. Okay, so we tried some of these systemic insecticides. Platinum, which is thiamethoxam, and Admire, which is imidacloprid, and you can see there really wasn't much effect there. Okay, with the control being 6.7, and you know, it's kind of interesting. At about this point in our work, we had an external review of the project, and so some people from the United States came under contract, reviewed the project, and one of the entomologists who was reviewing the project said, "Oh, this is an easy problem to solve. All you have to do is use one of these systemic insecticides and the problem goes away." Okay, well, the data showed that he was wrong, but it didn't seem to hurt him much because later on he became our associate dean for research, and now he's the chief scientist at NIFA. So apparently lack of knowledge doesn't hurt too much. So. Some observations about insecticides. In small plots, no benefit hardly at all. But one of the things I noticed as I traveled around various countries was that when we talked to farmers, and I would always ask them, have you sprayed insecticides? Whenever they were spraying insecticides, they had less disease pressure than where people were not spraying. Okay, and that really didn't fit with our general story that spraying insecticides isn't very effective for vectors. And what I think was going on there was, while a whitefly coming into the field that lands on a plant, he would feed on it and infect it, but if they die, they don't move to the next plant and infect it, so there's no secondary spread. And that's the best I can logically explain that, but it was very apparent that there was less disease when they were using foliar insecticides. Okay, but that's not going to solve the problem entirely, for sure. So then we come to probably the key part of the whole management system, and that is the host-free period. And here are the things that make this work. Only peppers and tomatoes are good hosts to the virus. The whiteflies are the only vectors, and the whiteflies don't pass the virus from one generation to the next. They only live for 30 days, and newly emerged whiteflies that don't feed on an infected plant will not carry the virus. Okay? So what we were going to do is then eliminate all the host of the virus for 2 months. Okay? So we go to 2 generations without any available hosts, and the idea was that we reduce the amount of virus in the system so that we could grow tomatoes successfully. Now, there's some problems with that. If you tell farmers, okay, you can't grow any tomatoes for 2 months, that's 2 months that they don't have any income. Well, then when they replant, it's 2 more months before they start harvesting tomatoes and peppers, and so now you got 4 months of no income. Now they can grow other vegetables on the same ground, so it's not like they're nothing, but they can't grow these vegetables. And then enforcement is a problem. If it's voluntary, there will be violators. If there are violators, it messes it up for everybody, okay, because the virus is in the community. In the Dominican Republic, they pass a law that certain months of the year you cannot have peppers and tomatoes in the field. That it's just a law. You can't do it. Okay. Okay, so what we had to do then was convince growers in an entire growing region to stop growing tomatoes and peppers during July and August. Okay, that's not an easy job. Okay, that's where Musa came in. Musa worked very closely with the extension people. He worked with the village chiefs and got them to agree that this is a good thing, that we need to stop growing tomatoes and peppers for that 2-month period. And we got greater than 95% of the people complying. And so it was a massive job. Musa, he's just a great guy and worked very, very hard to make this happen. Okay, so we the way it works then is we get rid of all the tomatoes and peppers during the months of July and August. Beginning September 1st, we plant the seed beds for the fall crop. At this point, we're going to use our best yielding varieties that have some resistance. Not necessarily the most resistant, but some resistance. And the reason we could do that is because the population of whiteflies are low at that point because it's the end of the rainy season, and hopefully the incidence of virus is going to be low as well. Then in January, when we plant the next crop, that's when the populations of whiteflies and virus is going to be high. That's when we're going to plant the most resistant varieties that we have available. So this is a graph showing what happened when we instituted the host-free period. And what we did here was we collected whiteflies from wherever we could find them, on other crops, on weeds, and so forth. And then we tested those for the presence of the virus. Okay, so here's our, I guess, Bill, I can't walk away, can I? Okay, here's our host-free period, July and August. You can see we had a lot of virus still present in July, but by August it went down to virtually nothing, and then it stayed low for several months. Okay, then it went back up to pretty high levels. The next year we did better because we had better compliance, so it was low in July. Again, almost zero in August, and then it went back up afterwards. Okay, so the host-free period was really effective at getting the virus populations down. Now, the fact that it rebounded the way it did tells us there are some other plants out there that are harboring the virus, because they got to get the virus from somewhere. But we really had no control over that, and I don't know what those are, to tell you the truth. So that's where they stand. So the host-free period was an effective way at getting the populations down to a level that we could live with. So what were the outcomes of this? The growers that we worked with adopted the resistant varieties and the use of the host-free period. Okay? As a result, their tomato yields dramatically increased. Okay? They went from virtually zero to very good yields, probably better than they were getting before the virus became a problem, because they now had improved varieties over the Roma. Okay. As a result, of course, they increased their income because they were able to sell tomatoes. Okay. Their standard of living improved because many of the growers of tomatoes were women. They spent the money on education and health care. Okay, not on a second husband. They wouldn't do that. So, but I was at an extension meeting once in Mali, and a bunch of men were sitting around, and so I asked them the question: when your wife grows tomatoes and she sells them, does that money go into the family kitty of money, or does she keep that for herself? And so, because I don't speak the same language they do, someone had to translate that for me. And so there's a pause, someone translates for me, and immediately every one of them laughed, because the answer was the same. No, the women keep the money for themselves. Okay, they don't let the men have it. Okay, and so that's why the money was spent well on education and health care. Okay, and then probably the final thing that happened was in 2011, Tom Cresswell from the PNPDL and I reviewed applications for Mali to start exporting tomatoes to the United States. And they were concerned about whether the viruses that we had identified would come over in the fruit, or if maybe the whiteflies would be transported here as well. And we both agreed that neither one of those were causes for concern, and so we approved those, and I think they got approval to start exporting tomatoes to the U.S. So we went from a situation where they could not even meet their own needs for tomatoes to they're exporting them throughout the region, and now even to the United States. So I think we really have accomplished what we're hoping to accomplish there. Okay. Now, Isufu Kolo, who was the plant pathologist from Niger who got his master's here at Purdue, he sent this message to us a few years ago. It says, imagine just a yield increase of 10 tons per hectare. This represents right now about $1 million cepha or $2,000. Okay, remember, these are farmers that have an income of $600 to $800 a year. Okay, so they're tripling their income. Okay, that's a lot of money, especially in an African village. People are interested in reducing poverty. This is an example, and the story should go out. Okay, so my take-home message is, what you do as an individual scientist may not seem like a whole lot. You know, my role in this was relatively minor, but you take a team of people, you put it together, you put it in action, and it has major consequences for the people involved. You know, if you're making $800 a year and all of a sudden you're making $2,500, your life has changed, you know, and changed for the better. Okay. Now, always have to tell who paid for this, and a lot of contributors there. Most of our money came from the IPM CRISP project. We did get a significant chunk of money from the USAID mission in Mali. We had to go visit them every time we went there to keep them informed of what we were doing, and eventually they came through with about $100,000 to support the project. Okay, and all these other people contributed as well. So let me go back to my original thought here. Why work internationally? Because it's a real opportunity to help people that need help. And so I got a series of slides here to show you some of the people that we helped. Okay. How can you say no to that face? So, okay. So, any questions?